About the digestion of protein, then absorption and metabolism in non-ruminant animals. So, animals, non-ruminant animals, they get their dietary protein in the form of some vegetable and animal protein feed ingredients. So we provide, you all of you know that we provide some ration to the animals, and in that particular ration, there will be different kinds of feed stuff or feed ingredients will be there, and some will provide energy, some will provide protein, some will provide vitamins, minerals like that. So mainly the vegetable and animal protein feed ingredients, they provide protein to the animals and to the non-ruminant animals also. And vegetable protein sources are refined oil cakes like groundnut cake, mustard oil cake, tea oil cake, or sesame cake, and soybean meal particularly. And the animal protein feed includes fish meal, meat meal, then meat cum bone meal, blood meal, feeder meal, slaughterhouse byproduct, etc. Different kinds of ruminant animals need different levels of protein in their ration, and it also depends on their age, body weight, physiological stage. That means if the animal is growing, then the animal may be pregnant or animal may be lactating, and depending on that also dietary protein level should be different in their ration. And productivity. Productivity means like if suppose lactating animals, in that case depending on the milk production, depending on the litter size, dietary protein level or requirement will vary, and that way we have to provide dietary protein to them. As for example, pig diet or swine diet should contain 12 to 20% crude protein, but depending on their physiological stage, requirement will be different. For example, at weaning, they need 20% protein. During finishing period, they need 14%. During gestation, 12%. Whereas in lactation, it should be increased to 18%. And second diet also should have different levels of protein depending on their age and depending on the production state or in case of layer. And in case of broiler, on an average, during the starting period need 22% CP, whereas during growing period need 20%, and during finishing period they need on an average 18% protein. However, we have some standard, we have some feeding standard for the non-ruminant animals, and we have to follow those standards. But these are the average values. And in case of equine or horse, they need approximately 12 to 14% CP in their ration. So in case of equine also, it will depend on their physiological state or stage. The stability of dietary protein is very important in non-ruminant animals. That means the quantity of protein we are providing through the ration is not important, but the amount of protein or amount of the end product of protein digestion available for absorption or animal is getting actually, that is important. And this is called the stability of the dietary protein. And which affect the stability of dietary protein in non-ruminant animals. And there some may be the biological factors like depending on the species, strains of animals, the stability of the dietary protein will be different depending on the physiological state also or status also. The stability of the dietary protein, the same kind of protein if you are providing, the stability may be different, and individual variations are always there. And depending on the environmental factor like temperature, pollution, noise, etc., also the stability of dietary protein may vary. And dietary factor include form, that means at what way we are providing the feed, odor, texture, and particle size. Particle size is very important. If it should not be too big, it should be too small. And if it is not appropriate, then also the stability of the dietary protein will be different, or it may be high, means it may be less. So since the digestibility is very important, so we have to consider different factors also while we think that how much quantity of protein we are providing, or we have to provide to the in the ration of the non-ruminant animals. So digestion mainly occur in the small intestine in case of non-ruminant animals, but there may be some digestion of protein in the stomach of the non-ruminant animals, because there will be secretion of some zymogens which will be activated to their active form, and there may be some digestion. But since the ingesta or feed will be for a very short time in the stomach, so therefore there will not be much digestion of protein in the stomach, and most of the digestion will occur in a small intestine with the help of different proteolytic enzymes. The digestion process of protein in non-ruminant involves denaturation of the dietary protein and activation of zymogens. Zymogens means whatever enzymes will be secreted in the stomach. So those enzymes will not be active, will not be in active form. So they will be zymogen. They should be activated, and it will be done by the hydrochloric acid, which will be secreted in the stomach. So they should be activated first. Then hydrolysis of the large peptide and the undigested protein in the small intestine by the pancreatic and intestinal proteases. So most of the digestion, as I told you, will occur in the small and large intestine. How protein is digested in the stomach? There are three secretions which are responsible for digestion of protein: hydrochloric acid, pepsin, and renin. Of course, renin is secreted in the young animals only. So renin is responsible for digestion of milk protein. It is not secreted in adult animal or growing animal. Only hydrochloric acid and pepsin will be secreted. Digestion of dietary protein in the stomach require not only active gastric proteases, that means the zymogens should be in the active form, but also hydrochloric acid, because hydrochloric acid is required for activation of the gastric proteases. Hydrochloric acid is synthesized by parietal cells of the gastric gland of the stomach, and it creates an acidic environment wherein there will be activation of the gastric zymogens. But there are many stimulatory as well as inhibitory factors which control hydrochloric acid secretion in the stomach, and we should know these stimulatory as well as inhibitory factors of hydrochloric acid secretion, because if there is no secretion of hydrochloric acid in the stomach, in that case there will not be any denaturation of the dietary protein, as well as the gastric proteases will not be activated, and thereby practically there will not be any digestion of protein in the stomach of non-ruminant animals. So hydrochloric acid is very important, and these are the stimulatory factor of hydrochloric acid secretions, and the factors may be endocrine in nature, maybe neurocrine in nature, or it may be nutritional or paracrine. And gastrin, it is a hormone secreted by the gastrin Z cells of the stomach, and it is the primary stimulus of hydrochloric acid secretion. And acetylcholine, which is released by the vagus nerve and enteric system. So it is also responsible, or it also stimulates hydrochloric acid secretion in the stomach. Then histamine, which is released by gastric enterochromaffin-like cells, and it is also stimulatory to the hydrochloric acid secretion in the stomach of non-ruminant animals. And there are many inhibitory factors of hydrochloric acid secretion. Like the number one is slow gastric emptying. That means when the ingesta or food will be for more time in the stomach, in that case also secretion of hydrochloric acid will slow down, and thereby what will happen, there may not be proper activation of the zymogens, and thereby this digestion of the protein may be affected. And high hydrogen ion concentration in the gastric and duodenal content. So this may also be means inhibitory to the hydrochloric acid secretion, because it will stimulate secretin. Secretin is another enzyme which is inhibitory to the hydrochloric acid secretion. Then nutritional factor like dietary protein, when dietary protein is very less, in that case also it will be inhibitory to the hydrochloric acid secretion. When there will be more fat intake, then also it will be inhibitory to the hydrochloric acid secretion. And feed particle size, when feed particle size will be too big or it will be too small, in that case also it will be inhibitory to the hydrochloric acid secretion in the stomach. And there are many gastrointestinal peptides which are inhibitory to the hydrochloric acid secretion, like secretin. It is released by the D cells in the duodenum and jejunum. Then gastric inhibitory peptides, it is also known as GIP, and also known as glucose-dependent insulinotropic peptide, which is released by the K cell in the duodenum and jejunum. So this factor will also inhibit hydrochloric acid secretion. Then vasoactive intestinal peptide released by gastrointestinal tract, brain, pancreas, heart, and many other tissues. And peptide YY, this is released by L cell in the ileal mucosa, the ileum part. Then somatostatin, this is a hormone secreted by the endocrine cell of the gastric epithelium. This is also a primary inhibitor of the hydrochloric acid secretion. So we should know, because since hydrochloric acid is related with the activation of the zymogen and thereby digestion of the dietary protein in the stomach of the non-ruminant animals. And the dietary factor I already told you. I think when there is food particle size or feed particle size is more, there will be more, or when there will be more feed intake, then there will be more distension of the stomach, and it will stimulate secretion of secretin, and which in turn will inhibit the secretion of hydrochloric acid. And high concentration of fat also activate this cholecystokinin release from the intestinal enteroendocrine eye cells in the duodenum, and thereby it will be inhibitory to the gastric hydrochloric acid secretion, or it will be in turn inhibitory to the digestion of protein in the stomach of the non-ruminant animals. So if these things are important, please try to remember. The dietary deficiency of protein, when, as I already told you, that when protein level is very less in the ration, it may be inhibitory to the gastric hydrochloric acid secretion. Then deficiency of zinc, B complex vitamin. It's like if like there is deficiency of protein, zinc, B complex vitamin, or if feed particle size is very large, then if it is small, very too small, in that case, it will affect, or it will act as an inhibitory to the hydrochloric acid secretion. So this hydrochloric acid secretion is important, I already told you, because like it is required for activation of the zymogens, which are responsible for digestion of dietary protein in the stomach of non-ruminant animals. So therefore, these factors, both stimulatory as well as inhibitory factors of hydrochloric acid secretion, is important. So now again I will discuss if it is connected. Then effect of this hydrochloric acid and the gastric proteases on protein digestion in non-ruminant. Okay. So how protein digestion occur in the stomach? So first of all, actually, dietary proteins will be denatured by the hydrochloric acid, and then it will be converted to large peptides for the actions of the active zymogens. So active zymogens will be pepsin A, B, C, and D, as well as renin. But renin will be available only in.
Sir.
Not listening.
Sir, we can't see the slides.
Huh?
So slides, it's not visible.
Okay, okay. Now it is visible.
Yes,
Okay.
sir.
Okay. Uh, so this is the mechanism. That means first the protein will be denatured, and after that it will be digested. Digested by the this gastric proteases, or you can say active zymogens, to large peptides. These zymogens are secreted. They are secreted by the Sheath cells of the gastric gland in the fundic and pilot region. And renin, it is a mammalian enzyme only. So in the bird, this renin will not be present, and renin is secreted only in case of young animals for digesting the milk protein. Then I already told you pepsins will break down peptide bonds, and after that there will be formation of large peptide. That means protein will be converted to large peptide. The next one of pepsins are inhibited by high pH and by pepstatin. So it is an hormone, and high pH means like when the pH will change to alkaline. That means when the ingesta or feed will move from stomach to the small intestine. So that the pH will be high. pH will change from acidic to alkaline. Then in that case, the action of the pepsin will stop. Regulation of secretion of gastric proteases in non-ruminant. So these are the factor. Like first one is dietary protein intake. When dietary protein intake will be less, in that case, the amount and activity of the proteases may be less. Then gene expression in response to enteral feeding and glucocorticoids. So these two may also be responsible for secretion of gastric proteases in non-ruminant animals and their action. Then release of acetylcholine from the vagus nerve. Then acid concentration in the stomach. So this is also one of the stimulatory factor for release of gastric proteases. And secretion of cholecystokinin, gastrin, and other gastrointestinal peptides. I have already discussed about those gastrointestinal peptides. They will be stimulatory for the secretion of the HCl as well as gastric proteases. But this somatostatin, this hormone, and peptide YY inhibit the secretion of both gastric proteases and gastric acid. Digestion of protein in the small intestine. So in the small intestine, protein will be digested with the help of pancreatic proteolytic enzyme as well as enteric proteolytic enzymes or intestinal proteolytic enzymes. But here, pancreatic proteolytic enzymes will also be secreted in inactive form. So they will be activated in the duodenum, and after that, there will be digestion of protein. But proteolysis is limited in duodenum because of its short length. So as compared to ileum and duodenum and ileum, this length is short. So therefore, for short duration only, it will be present, and therefore there will be limited digestion of dietary protein in the duodenum. When chyme moves into the jejunum, most proteolysis takes place due to its long length and high protease activity. So that means most of the digestion will occur in the jejunum. Continuous digestion of protein and peptides can also occur in the ileum if their degradation is not completed in the jejunum. So that means throughout the small intestine there will be digestion of dietary protein, but it will be proteolysis will be more in the jejunum as compared. Okay, from here today I'll start. Okay, regulation of secretion of gastric proteases in non-ruminants. So I think the previously other things I have discussed. So here secretion of gastric proteolytic enzymes will be controlled by many factors, and mainly there are 5 factors which are responsible for stimulation of the secretion of gastric proteolytic enzymes, or you can say gastric proteases and peptidases. So number 1 is dietary protein intake. So there should be sufficient protein intake. Then only there will be sufficient or optimal secretion of gastric proteolytic enzymes. When protein intake will be very low, that will be inhibitory for the gastric protease secretion. Then number 2 is gene expression in response to eternal feeding and glucocorticoids. When we provide food through tube or IV in disease condition, that means when the animal is unable to consume food, then also there will be stimulation of gastric proteolytic enzyme secretion. And glucocorticoid therapy also will be stimulatory. Then third one is the release of acetylcholine from the vagus nerve. When there will be food in the stomach, then it will stimulate the secretion of acetylcholine, and it will be stimulatory for the secretion of gastric proteolytic enzymes or gastric proteases. Acid concentration in the stomach, it is also important, because then only the zymogens, or you can say that gastric proteases will be activated. Then fifth one is secretion of cholecystokinin, gastrin, and other gastrointestinal peptides. So this will be stimulatory for the secretion of gastric proteolytic enzymes. But the somatostatin, this is one. The somatostatin and peptide YY, these two are secreted by the epithelium. So these two will be inhibitory for the secretion of gastric proteolytic enzymes as well as gastric hydrochloric acid or gastric acid. And how proteins are digested in the small intestine? Here, whatever will be left or whatever will escape digestion in the stomach, those will be digested with the help of pancreatic proteolytic enzymes as well as enteric or intestinal proteolytic enzymes. But their action will only be in alkaline medium. There are three different parts, all of you know, duodenum, jejunum, and ileum of the small intestine. But since duodenum is very short, then because of that, actually proteolysis will be limited. And moreover, since for the action of pancreatic proteolytic enzymes as well as intestinal proteolytic enzymes, there is need of alkaline environment. But immediately after reaching the chyme from the stomach, it will not be totally. There will be some acidic condition in the chyme. So therefore, this might also be another reason why actually proteolysis is limited in the duodenum, and it will be very less as compared to other part of the small intestine. Chyme moves into the jejunum, most proteolysis will be taking place, because here the environment will be totally acidic, because everything will be mixed up and most of the digestion of the protein will occur in the jejunum part of the small intestine. Then, but if it is not completed, or if some portion of the protein left digestion, then that will be digested in the ileum part of the small intestine. Acinar cells of the pancreas secrete all the proteolytic enzymes, and they will be secreted in inactive forms, or we can say they will be secreted as proproteases or zymogens. They will be trypsinogen, chymotrypsinogen, and procarboxy peptidases. There are different types of trypsinogen, chymotrypsinogen, ABC as well as elastase, and carboxypeptidases will be of two types, A and B. And enterokinase secreted by the intestinal epithelium will be responsible for stimulation, or activation of the trypsinogen into trypsin, and trypsin in turn will activate other pancreatic zymogens or pancreatic proteolytic enzymes. Bile acid and cholecystokinin stimulate the production of enterokinase and proteases by the pancreas. So these two is also necessary, that means for stimulation of the secretion of the pancreatic proteolytic enzymes, as well as activation of the pancreatic proteolytic enzymes by the enterokinase. Intestinal epithelium or intestinal cells also secrete proteases as well as oligopeptidases. So this will also be responsible for hydrolysis of dietary protein. And aminopeptidases, they will hydrolyze protein or large peptide from the nitrogen terminus. And oligopeptidases, they will hydrolyze peptide bond in oligopeptides consisting of 2 to 20 amino acid residue. Then how amino acid will be absorbed? So this end product of protein digestion will be amino acid ultimately, and how they will be absorbed from the small intestine. There are two ways by which actually amino acid absorption can occur in the small intestine. And the first one is transcellular absorption, and which is generally present in mature animals. And second one is paracellular absorption, and which is normally present in young animals. Paracellular absorption is only available or only present in the young animals, because this paracellular absorption means this absorption in between through the tight junction of the enterocytes, whereas transcellular absorption means through the intestinal epithelial cells or through the enterocyte of the epithelium. And this means immediate after birth, you know that this colostrum, it contains immunoglobulin, and immunoglobulins are absorbed as such from the small intestine of the neonate or young animals. So that absorption pattern will be paracellular absorption of protein. Okay, and the gut closure means like after 24 hours after birth, actually that immunoglobulins will not be absorbed as such, and that particular phenomenon is known as gut closure. And after that, what will happen? After that, they will be digested, and they will be digested to amino acid, and then in the form of amino acid, they will be absorbed. So you remember that paracellular absorption of dietary absorption of end product of protein digestion is present only in young animals, means before gut closure, and after that all the absorption will be transcellular. That means via the intestinal epithelial cells or enterocyte, and there will be two mechanisms. One will be active absorption, and another will be passive absorption. That means active absorption means because of concentration gradient, and passive absorption means through some compounds or with the help of some other compounds or mediators. In protein digestion in large intestine, suppose if some protein is left, then those protein will be digested in the large intestine. But here, like in the large intestine, you know that there are three different parts: cecum, colon, and rectum. And in some non-ruminant animal, cecum is well developed, but in most of the non-ruminant animals, cecum is not well developed. So in the cecum, there will be microorganisms like rumen of the ruminant animals, and these microorganisms will be responsible for hydrolyzing the leftover protein or protein escaping digestion in the small intestine as well as stomach. But like what happened? So digestion of protein by the microbes or by the bacteria present in the large intestine will result production of amino acid as well as ammonia gas. And after that, this amino acid and ammonia gas may be utilized by the bacteria or microbes for synthesizing their own body protein. And some ammonia or amino acid may also go to other part of the large intestine. But here, like for getting microbial protein or microbial amino acid by the animal, the microbes should be digested. Okay, microbes should be digested, and after that, microbial protein should be digested. But you know that from cecum, after cecum, there will be only colon and rectum. So in the colon part, almost there will not be any secretion of digestive photolytic enzymes. So there will not be any digestion of protein in the colon. Sorry, there will not be any digestion of protein in the colon, as well as means microbial protein will not be digested. But if there is some amino acid, that amino acid, of course, may be absorbed to the colon to some extent. But the amount of free amino acid represents less than 1% of the hindgut fermentation product output. So that means it will be very less. It will be very, very insignificant. That means the available amino acid after digestion in the cecum of non-ruminant animals will be very, very less, and therefore it will be very insignificant. So it is written, however, through the epithelial cells of the cecum and colon is much lower. However, absorption through the epithelial cells of the cecum and colon is much lower for amino acid and peptides, and the large intestine cannot absorb intake protein. So therefore, hindgut fermentation of dietary protein has a limited role in providing amino acid to the host animal. So this is the thing in case of non-ruminant animals, although there may be synthesis of some microbial protein in the hindgut, in the cecum of some non-ruminant animals. So absorption, I already told you, there are two absorption mechanism. Transcellular absorption will be there via active and passive transport to the enterocyte. Protein metabolism in non-ruminant animals. So you know that animals utilize protein for synthesis of the body protein, that means muscles of the animals. But when there will be deficiency of carbohydrate and fat, which are the main sources of energy for the animal, amino acid may also be utilized for energy production or as a source of energy. And that particular mechanism or metabolism is known as gluconeogenesis. That means production of energy from sources other than carbohydrate. Okay. Amino acid metabolism, it involves two different steps. One is deamination and another is transamination. Actually, this degradation mainly takes place, or this catabolism or protein metabolism mainly takes place in the liver, and to some extent in the kidney. But muscle tissues are electrically inactive. That means in a muscle there will not be any catabolism of protein. So first stage of is oxidative degradation of amino acid. That is removal of amino group by one of the following pathway. One is oxidative deamination, and another one is oxidative transamination. So in transamination, amino group is transferred to the alpha carbon atom of keto acid, resulting in production of another amino acid and keto acid. I think you may know this transamination and deamination. It may be discussed in the biochemistry. So here, of course, ultimately means after this through this deamination and transamination of the amino acid, ultimately there will be production of acetyl coenzyme A. So that acetyl coenzyme A will enter into the TCA. After that, it will be metabolized by TCA cycle for production of energy. That means end product of amino acid catabolism via oxidative deamination and oxidative transamination is then alpha ketoglutarate. After that, it will be means metabolized to acetyl coenzyme A, and this acetyl coenzyme will be metabolized by TCA cycle. So here, for some amino acid like tryptophan, leucine, and isoleucine, acetyl coenzyme A may be produced directly. But for these amino acid, namely alanine, glycine, serine, threonine, and cysteine, acetyl coenzyme A will be produced via pyruvate means when there will be oxidative deamination, transamination. After that, there will be production of pyruvate, and then acetyl coenzyme A will be produced. Then for this amino acid, phenylalanine, tyrosine, leucine, lysine, and tryptophan, acetyl coenzyme A may be produced via acetoacetyl coenzyme A. First of all, there will be production of acetoacetyl coenzyme A, then there will be production of acetyl coenzyme A, and then it will enter into the TCA cycle. Other amino acids are degraded by pathways yielding products like alpha ketoglutarate. So alpha ketoglutarate is a metabolite of the TCA cycle. Oxaloacetate, it is also a metabolite of TCA cycle. Fumarate, it is also one metabolite. Succinyl coenzyme, it is also another metabolite of TCA cycle. So when there will be formation of acetyl coenzyme A, sorry, alpha ketoglutarate, so it will enter in this particular form into the TCA cycle, and after that there will be the listening now. Okay, okay, I'll continue now. Okay, then ammonia is produced. Ammonia is also produced from catabolism of amino acid, and this ammonia uh, will be excreted as urea, and mainly in mammals, as well as amino acid in birds. So how they are formed? So I'll discuss now. So urea cycle means this urea will be synthesized from ammonia via urea cycle, and here there are two stages. In the first stage, there will be formation of carbamoyl phosphate from carbon dioxide and ammonia. So here, this carbamoyl phosphate synthesis will be required, and there will be involvement of 2 ATP. That means 2 ATP will be required, and this enzyme will be required, and there will be formation of this particular compound. Then, second stage is reaction of carbamoyl phosphate with ornithine to start a cycle of reaction in the production of urea. So after that, this cycle, that means from ornithine, ornithine plus carbamoyl phosphate will produce acetylene, then arginosuccinate will form, and after that arginine, then there will be yield of urea, and there will be production of ornithine. But here there will be involvement of various enzymes, and you try to remember these two stages of urea cycle. Okay, first stage is this formation of carbamoyl phosphate, and then second stage is reaction. Cycle is not present. Urea will be synthesized, uric acid will be synthesized, and this involves incorporation of ammonia into glutamine by reaction with glutamate. Okay, so this ammonia will react with glutamate, and there will be production of glutamine. Glutamine is then involved in series of reaction yielding inosinic acid. Okay, and then inosinic acid will undergoes to xanthine oxidate mediated oxidation to give xanthine and uric acid. Okay, that means ultimately uric acid will form, and in that particular form, the nitrogen will be excreted. Protein synthesis there are different steps of different ways of protein synthesis in animals, and some of the amino acid will be synthesized, but all of you know that most of the amino acids are dietary essential for the animal, and they are known as essential amino acid. And there are 10 essential amino acid in case of monogastric animals, and 11 essential amino acid in case of bird. Okay? And glycine, along with other actually 10 essential amino acid, is required by the bird, and therefore there is need of 11 essential amino acid in case of bird, whereas non-ruminant animals need 10 essential amino acids in their diet, because these essential amino acids cannot be synthesized, but other amino acids can be synthesized from amino acid of the dietary origin. So this process of protein synthesis, it involves 4 different stages. Number 1 is activation of individual amino acids. Then number 2 is initiation of peptide chain formation. Then chain elongation and chain termination. So these things, from the nutritional point of view, actually not much important. I think these things will be discussed in details by the biochemistry people. So from the animal nutrition point of view, we just remember the steps involved in the protein synthesis in animals. Okay, we just remember only the names. Okay. So this is all about the digestion, absorption, and metabolism of protein in non-ruminant animals. Okay. So here you remember protein digestion is like in the mouth there will be any digestion of protein, because there will be any synthesis of or there will be any release of proteolytic enzyme in the mouth of the non-ruminant animals. In the stomach, there will be secretion of pepsin and renin, pepsinogen and renin, pro-renin actually. Zymogen will be secreted first. But anyway, this pepsin and renin will be the proteolytic enzyme in the stomach. Renin is present only in the young animal for digestion of milk protein, but this pepsin is present in adult animal. So there will be some digestion. But here also, like the food will stay only for a very short duration in the stomach. So therefore, there will not be much digestion of protein. Only there will be formation of large polypeptide from protein in the stomach for hydrolysis by pepsin. Okay. After that, when this ingest or chyme will go into the duodenum, so in the duodenum there will be release of this pancreatic proteolytic enzymes. But in the duodenum also there will be much digestion because it is very short, and moreover the chyme will not be fully alkaline, because immediately after entering from the stomach into the duodenum, still the chyme will be in acidic form, and because of that actually that this proteolysis or hydrolysis by the pepsin will still goes on. But when it will mix properly with the secretions in the duodenum, then when it will be completely alkaline, then there will be action of the pancreatic proteolytic enzymes. But most of the protein digestion will occur in the jejunum part of the small intestine with the help of both pancreatic proteolytic enzyme as well as intestinal proteolytic enzymes. You just try to remember things. Then if there is if some proteins left or if some polypeptide left, then those will be digested in the ileum part of the small intestine. And after that, still if there is escape of some protein, dietary protein in the small intestine, it will be hydrolyzed by the microorganisms or bacteria in the cecum of the large intestine, and there will be formation of amino acid as well as ammonia, and this amino acid and ammonia will be utilized by the bacteria for synthesizing their own body protein, which is also known as microbial protein. Okay, and there may be some amino acid, free amino acid also in the cecal or cecum content. Okay, so after that, but it is very less than 1%. Okay, I have already shown you. But since after cecum there is colon and rectum only, so ultimately what will happen for getting amino acid, for getting microbial protein by the animal, there should be digestion of microbes, whatever, or bacteria, whatever will be present in the cecum, but they will not be digested. So therefore, animal ultimately will not get microbial protein. Or moreover, absorption of protein or amino acid will be very less in the large intestine. So therefore, hindgut fermentation or hindgut digestion of protein in case of monogastric animals is not so significant. Okay. Then metabolism, mainly proteins are or amino acid will be utilized by the animal for synthesizing their body protein or muscles. But when energy is less or when energy is deficient in the diet or when carbohydrates is less or fat is also not present, in that case, the animal may utilize amino acid for production of energy. So how the animal will utilize amino acid for production of energy? There will be deamination as well as transamination, and after that there will be production of acetyl coenzyme A. And this acetyl coenzyme A will enter into the PCA cycle, and there will be production of energy in the form of ATP. So for some amino acid, the acetyl coenzyme will produce or will produce directly, but for some amino acid there are some other intermediate metabolite, and but ultimately what will happen, whatever metabolite will form, and they will be metabolized by a PCA cycle for yielding energy in the form of ATP. So this way, very briefly, you just try to remember, and this will be important from the examination point of view. Okay. Do you have any question? Hello? Are you listening?
No, sir.
So if any question you can tell. And this thing I think you got in other course also, I think, is not it? In physiology it was discussed or not? Hello? Huh? Hello? So anyway,